You're welcome, guys. So the third MCQ reviews in amino acid metabolism. Question 1 says, which enzymes are responsible for producing the direct donors of nitrogen into the pathway producing urea? So the answer to this is aspartate aminotransferase and carbamyl phosphate. This provides aspartate, and carbamyl phosphate synthesis provides the carbamyl phosphate. So the carbamyl phosphate synthesis reacts the ammonia plus bicarb and to form carbamyl phosphate. And this and the enzyme that does this is CPS, carbamyl phosphate synthesis, which is also activated by N-acetyl-glutamate, as we've said. And ATP is used up here, 2 ATP reduced to ADP plus PI. So to go back to the lectures now. So the answer is aspartate amino transaminase AST and carbamyl phosphate synthesis CPS. Question 2. 2 days after full-term normal delivery, a neonate begins to hyperventilate, develops hypothermia and cerebral edema, and becomes comatose. Urinalysis reveals high levels of glutamine and orotic acid. BUN is normal. Which enzyme is most likely deficient in this patient? The answer is mitochondrial ornithine transcarbamylase. So we are unable to react carbamyl phosphate with ornithine to form citrulline. So the enzyme that does this is mitochondrial ornithine transcarbamylase. So this enzyme is not working. This reaction will not work. So, and there will be orotic aciduria because the carbamyl phosphate would leak out of the mitochondria and go into the cytoplasm and go to the pyrimidine pathway. So it's in the pyrimidine pathway, it gets converted to orotic acid. So it bypasses the activity of carbamyl phosphate synthesis 2. So this one is carbamyl phosphate synthesis 1. I didn't state the specific one then. So question 3 now. A newborn becomes lethargic and drowsy 24 hours after birth. Blood analysis shows hyperammonemia, that's the first abnormality here, coupled with orotic aciduria. This individual has an enzyme deficiency that leads to an inability to produce which of the following? The answer is citrulline. So citrulline is not produced in this patient. Considering the patient in question 3, orotic acid levels are high in this patient due to which of the following? We already said it in question 2. It bypasses carbamyl phosphate synthesis 2. Considering the patient discussed in the last 2 questions, that's question 3 and 4, a potential treatment for the patient is a supplementation with which of the following? We can use arginine and benzoate. So arginine can be used, and benzoate will react with glycine to form hypuric acid. You could refer back to our lectures. The amino acid that does not participate in transamination, question 6 now. We have lysine, threonine, proline, and hydroxyproline. They do not participate in aminotransferase reaction. So the answer here is lysine. So for question 7 to 10, as simple orientation, this is just the TCA cycle or the citric acid cycle. And don't forget the mnemonics we gave for citric acid cycle. We said, can I keep selling skills for money, Dr. Onos? And I said the answer to you guys is obviously yes. This is the cell's way of selling skills and making money in form of ATP. So the can there, the C is citrate, sorry. The I is isocitrate, which is this option C here. The K here is alpha ketoglutarate. The S here is succinyl CoA. So that's option E here is succinyl CoA. So let me write it well. Succinyl CoA. This is succinate. The F here is fumarate. The M here is malate, and the G here is oxaloacetate. And of course, if this is oxaloacetate, this B here should be acetyl-CoA, and this should be pyruvate. Pyruvate can go to acetyl-CoA with the activity of the enzyme pyruvate dehydrogenase, and pyruvate to oxaloacetate by the enzyme pyruvate carboxylase. So let's go and answer the questions now. Question 7 says an obligate activator of hepatic pyruvate carboxylase in the post-absorptive phase, and the answer should be acetyl-CoA. So option B should be the correct option here. Option B for question 7. So question 8 says product formed by adenylosuccinate lyase during urea synthesis, and the answer is fumarate. So that should be option F. Question 9 says substrate and energy source for synthesis of delta amino levulinic acid in heme pathway, and that's succinyl-CoA. So the answer here should be E, succinyl-CoA. Converted to glutamate in a reaction requiring the coenzyme form of pyridoxine. Of course, that should be alpha ketoglutarate, option D. And the coenzyme there is vitamin B6 pyridoxal phosphate. And the enzyme that carries out those reactions are amino transferase reactions. So that's it for 7 to 10. Question 11 says, a research scientist is studying biochemical reactions that takes place in the liver. He cultures hepatocytes in a growth media enriched with glutamate labeled with nitrogen isotopes. After some time, he finds that nitrogen isotopes are transferred to oxaloacetate, forming aspartate in the process. Which of the following substances is most likely involved in this reaction? We already said pyridoxal phosphate from pyridoxine, so the answer is option D. Question 12 says, as part of the experiment, healthy volunteers undergo a 12-hour fast and then drink a solution containing radiolabeled alanine. Consecutive blood samples are drawn every 15 minutes for the next 3 hours. Initial blood samples detect the radiolabeled alanine, but analysis of later samples shows that the radiotracer is present in blood, primarily in the form of glucose. Before alanine can be converted to glucose, its amino group is transferred to which of the following? The answer is alpha ketoglutarate to form glutamate. So that's exactly how the process is, and in turn the alanine gets converted to pyruvate, which then goes for gluconeogenesis because they've been fasting for 12 hours. Don't forget that. Question 13 says, a 4-day-old boy born to a 23-year-old woman is brought to the office for evaluation of poor feeding and vomiting. The pregnancy was uneventful and the mother had a normal delivery. Family history is non-contributory. The patient's temperature is 37.2 degrees Celsius. Blood pressure is 60/30 millimeter of mercury. Pulse is 1/10 beats per minute. And respiration is 56 cycles per minute. Physical examination reveals a lethargic newborn with exaggerated deep tendon reflexes and clonus. Further investigation reveals that the patient has an inherited condition that results in impaired transport of ornithine from cytosol to mitochondria. This child likely has HHH syndrome, which is also called hyperammonemia, homocitrullinemia, and of course he has hyperornithemia. So those are the three conditions of HHH syndrome. So nutritional restriction of which of the following substance can improve the patient's condition is to restrict proteins in general. So reduce the protein levels and the urea cycle pathway would reduce and the production of ornithine would actually come down too. So that's one of the treatments for patients with HHH syndrome. Question 14 says, a newborn becomes lethargic and drowsy 24 hours after birth. Blood analysis shows hyperammonemia but no acidosis. The newborn was presented with friable tuft hair, which enzyme could be deficient. Now, if you say friable tuft hair in urea cycle is pathognomic of adenylosuccinate lyase deficiency, which would lead to adenylosuccinic aciduria. So when the patient has adenylosuccinic aciduria due to deficiency of adenylosuccinate lyase, you see this classical presentation of friable tuft hair in the baby. Question 15. A newborn presented with symptoms of hyperammonemia. Blood analysis done showed no acidosis, no elevated levels of any specific amino acid. what will be your next line of investigation? I'll likely do a urinary or orotic acid level test. So I'll obtain urine orotic acid levels. So that will be the answer. You can refer to the lecture notes for more details about the investigations done during hyperammonemia. So thank you guys.